DNA replication is the process in which DNA is copied for the later synthesis of proteins. DNA replication occurs inside the nucleus, where DNA is stored, and requires an enzyme to be catalyzed. It occurs through two processes, transcription and translation. Transcription is the first part of DNA replication, and it occurs within the nucleus just like the entire process of DNA replication. Transcription is essentially the process in which a strand of mRNA is made complementary to a strand of DNA, using the original strand of DNA. It happens when the enzyme RNA polymerase splits the DNA's pre-existing complementary nucleotides, therefore splitting the DNA strands into two separate ones. RNA polymerase does this by binding to a region of the gene called the promoter that tells the DNA to unwind. From here, RNA polymerase is able to read the DNA and later make a strand of mRNA with a complementary sequence of bases. This is called initiation. The next step is the actual addition of the nucleotides to the one DNA strand to make one strand of mRNA. The enzyme reads the one strand of DNA and pairs complementary nucleotides to form the mRNA. The complementary bases are as follows: C to G, T to A, or U to A in RNA. There are three types of RNAs. mRNA carries genetic blueprints from the DNA in the nucleus to the cytoplasm, as mentioned in transcription. rRNA aids in the formation of ribosomes. And then tRNA brings amino acids to ribosomes to join them together and form proteins, which will be mentioned during translation. Transcription ends with termination, where the mRNA strand is done and removes itself from the DNA strand. In between transcription and translation, there are a few processes a new strand of mRNA may go through. Editing changes nucleotides within the mRNA that can change the outcome of a protein, adding stop signals in a specific place, for instance. Splicing is the removal of introns that do not code for proteins, but instead for other material, so that the mRNA only consists of extrons, which are regions that code for proteins. Then polyadenylation adds a protective tail to the mRNA that also signals the end of mRNA. Then translation begins. The strand of mRNA meets with the ribosome outside the nucleus. The ribosome makes use of the sequence of codons within the mRNA and uses it to send for tRNA that brings amino acids to the ribosome in the correct sequence in order to create a secondary strand. Codons are groupings of three nitrogenous bases put together to form a code, the genetic code. A codon encodes one amino acid. 64 different codons can code for 20 different amino acids. Codons then also let the ribosome know where to start and stop calling for tRNA to bring in more amino acids. Anticodons are the complements to codons already on the mRNA strand. tRNA is a strand of RNA nucleotides that recognizes a specific amino acid and provides anticodons to complement codons during translation. Each tRNA is specific to a certain codon. A polypeptide chain, or protein, is formed by the continuous matching of anticodons to codons on the mRNA strand. To wrap things up, DNA replication is the process in which DNA is copied to help form a protein specific to the cell. DNA replication occurs by transcription and translation. Transcription occurs when RNA polymerase binds to the Tata box within the promoter region on a strand of DNA, catalyzing the unwinding of the DNA's helices. RNA polymerase then advances to one of the strands of DNA and brings in the complementary base of every nitrogenous base included within the section of DNA being copied. From here, a strand of mRNA is created of the complementary nitrogenous bases, and it is ready for translation once exiting the nucleus. The term complementary is based on the following: C to G, T to A, G to C, A to T, or A to U in the case of RNA. Each one of these letters stands for a nitrogenous base. Translation is the process in which a ribosome reads the mRNA and sends for tRNA that calls for the mRNA's complements to create a chain of amino acids, also known as a polypeptide chain or a protein. More detail is gone into on each individual slide.